So, blood is one of the connective tissue and it is also called a special connective tissue or fluid connective tissue, because it is a circulating fluid. As we know, in the developed human beings or these developed animals, the blood flows within a closed system. It is the circulatory fluid, it is within a closed system. In unicellular organisms, nutrients and waste materials can be exchanged just by diffusion from outside to inside. Unicellular organisms, right? Unicellular organisms just by diffusion. What is diffusion, osmosis, the concepts of these will be discussed just a bit in detail in general physiology. So, you have an idea about diffusion, but in developed animals it is possible. Is it possible to exchange materials like that, whatever nutrients, waste products? No. In multicellular organisms, so it is the circulatory fluid which circulates within a closed space, and this is called the vascular space, right? This is simple, pretty simple till now. In the vascular space, blood flows, the liquid that flows is blood, okay, and it is providing the body with nutrition, oxygen, whatever, nutrition, the functions of the blood, every transport functions is the functions of the function of blood is mainly this transport function, that transport the nutrients and waste material, nutrients and oxygen to the tissues and carry the waste materials from the tissue so that they can be excreted from the body. So, in the adults, what is the volume of blood present? Normally 5 to 6 liters. Okay, in the females it is somewhat less, and among this blood is composed of a fluid part which is called the plasma, and rest is the cells, also called the formed elements of blood. All these things are not in very much not these are for, in these days these are for the MCQs. See, the blood is slightly alkaline. What is the what do you mean by acidic pH and alkaline pH? Concept of pH will be discussed in general physiology. So, you have this pH is just the basics, you know, pH is the negative logarithm of the hydrogen ion concentration in the body. So, that is, or anywhere, or any chemical composition. Here we are talking of the human body. So, it is slightly alkaline, it's slightly the specific gravity is higher than water, but the viscosity is much higher than water, like 4 to 5 times of that of water. So, this is a fluid connective tissue composed of plasma and the formed elements. This is what is just basic introduction, what all this you know. So, this is again, so what is this? A tube like structure. What is this? This looks like a test tube, okay, blood vial, okay. So, this looks like a test tube. So, there is a tube called the Winroes tube. So, all these names there will be various names in these discussions. So, whenever you discuss this any topic on medicine, there will be, medicine means the medical curriculum as a whole, there will be various names. These names are according to the names of the scientists or the names of those individuals who established this. So, this Winroes is the name of an individual who described this Winroes tube. This Winroes tube, if blood is allowed to, blood is drawn, and then a certain volume of blood is kept in the tube. Then what happens? Then the cells settle down. Cells settle down, and there is a layer of clear fluid above, and this takes some time. In 1 to 2 hours, in 30 minutes, 1 hour, 2 hours, this will be very clearly, that means it can be demarcated. So, the blood cells, the red blood cells settle down at the bottom of the tube, and the clear fluid is above, and between them is what is classical puffy coat, and that contains the white blood cells and the platelets. So, this is the concept of something called the erythrocyte sedimentation rate, which we will discuss later, okay, and we will also show it to you in the physiology lab, okay, physiology laboratory, that red blood cells, they will settle down, and these red blood cells make up around 40%, around 40 to 45% of the total blood volume, red blood cells. So, if blood volume is in 100 ml blood, how much will be the total volume, not the number, this is the volume of the RBCs. Total volume of the red blood cells is around 40 to 45%, and these theoretical knowledge you will require in various areas. This theoretical knowledge, that what is the percentage of red blood cells, white blood cells and platelets, as you know already, these are very, if we compare to red blood cells according to number, these are very less. So, white blood cells, we theoretically we describe the number in the blood cells in per microliter of blood. So, if we would try to state this number, not describe, the correct word should be state this number in the whole of the human body, then how much blood is there? 5 to 6 liter, then it would become a huge number. So, it is described in microliter of blood. Micro means 10 to the power minus 6. So, per microliter of blood we express this number. So, what is the number of RBCs per microliter of blood? It is 5 to 6 million, 4.5 to 5.5 million as per standard textbooks, but WBCs are in thousands. Platelets, it is 1.5 to 4, WBCs is in thousands, 4,000 to 11,000. So, whenever you state a number, in most cases, whenever you state, most means in 99% cases, whenever you have to state something, you state it like this, that there should be a range. You have to state a range, not a particular number. Okay. So, it can't be the same for everybody. In medical science, it is not like 2 plus 2 is 4. So, that is not definite. So, it will be 4,000 to 11,000 is taken as the normal range for WBCs. It is in microliter, and in some texts, who have the students who have still now attended the practical sessions, we have already discussed microliter or cubic millimeter. It is the same thing. One is standard international system, and another is in CGS system, microliter or millimeter cube. If somebody does not get it, please raise your hand and ask. Although lecture sessions are primarily meant to be one-sided, the demonstration sessions are more interactive, but if you can't understand anything, so these units you should be acquainted, because these units will be needed repeatedly. So, for the examination purpose, and you have to read the reports also, you have to read reports. So, even in some cases in the western countries, they are putting it in per liter of blood. So, per liter of blood, it has to be multiplied by 10 to the power 6, it is written as into 10 to the power 6, okay, or per dl of blood. So, there will be some parameters will be there, which will be written per deciliter of blood. So, you have to just convert, like amount of hemoglobin we express in per deciliter of blood, the mass of hemoglobin per deciliter of blood. The blood cell counts are usually described in per microliter of blood, and plasma is greater than 50%. Classically it is told as 55%, but again it is not that fixed. It can't be a fixed, it is a range, but classically it is taken that it is 55% of the total volume, which is the blood plasma, which is the fluid portion of the blood. Okay. And the blood plasma, we will come to the composition of the blood plasma, but the cells of the blood are RBCs, WBCs and platelets, red blood cells, white blood cells and platelets. Red blood cells white blood cells are again classified as whether they contain granules or not. Those are granulocytes which contain granules, and agranulocytes which contain which do not contain granules, and for who are still freshers compared to the others who have just joined, we are doing this now in the hematology lab as well. Okay. Hematology lab, you have to count the number of, you have to identify cells under the microscope, different types of white blood cells. After staining a blood film, you have to identify the different types of white blood cells, differentiate them from red blood cells, count the number of white blood cells. You have to count at least 100 cells and express the different types of white blood cells as a percentage of the total number. That means, if you count 100 cells, how many of them are the different types of granulocytes, namely the neutrophils, eosinophils, basophils, and how many are lymphocytes and monocytes. Among them, we will discuss again, but basophils are very rare. Basophils are very rarely found. Out of 100 cells, even there, if we count 100, 200 cells, even there may be no basophil. These are very rarely found, but these are there in the body, but under the microscope, counting only few cells like 100 or 200 blood cells, it is difficult to get a basophil. Okay. But those are, basophil is also one of the granulocytes. So, when you read about the structure of the WBCs, then again we will discuss. So, this is just an overall that what is the composition of blood, plasma and the cells. This is physiologically. That means, when the physiological, whenever you use the term physiological, we are considering that the body is devoid of, it is not diseased. So, it is just the normal functional characteristics of the body we are discussing whenever the term physiological is told, and whenever we are thinking of a diseased entity, diseased body, then the term used is pathological. So, under physiological conditions, blood contains plasma and these different types of cellular components. Now, why are the cellular components called the formed elements of blood? Why are they called formed elements? This left hand side, say I have combined two images. So, these left hand sides just illustrates the different cell types, and the description is whatever is found under the microscope, we will discuss it in the hematology lab. So, and also the theoretical portions in these lecture sessions, but hematological lab, those who have already seen the cells under the microscope and well appreciate it, that what are the structural features by which we will differentiate the RBCs, WBCs, and among the WBCs the different types of WBCs. Platelets are still not the stains which we are using, platelets are still not so, I think you may not have seen platelets till now, but platelets can still be found. So, here we are staining it with a stain called the Leishman stain. Okay. So, by staining we can identify the different types of RBCs and WBCs, and there are the description of how we will identify the different types of cells, which we will do it in the hematology lab itself. And why are the cells called the formed elements of blood? Because firstly they are non-fluid components. Fluid components are not that well formed in structure, they are non-fluid components. Okay. These are non-fluid components having a definite structure, and actually they are formed from multipotent stem cells. We will discuss about hemopoiesis in a bit detail in the coming classes. So, they are formed from multipotent stem cells and they have definite structure. So, they are formed elements of blood. And why instead of cells the term formed elements is better? Because at least one of the components of blood, that is platelets, are not whole cells, but cell fragments formed from their precursor. What is the name of the precursor of the platelets? Megakaryocytes, right? Like all these blood cells, when they are formed from the stem cells, they are structurally disintegrating, and those have names. So, they are called the precursors. Precursors are the elements from which the next step in a biological process will be formed, next cell. So, the precursors of platelets are called megakaryocytes. So, platelets are actually not whole cells, but they are formed from fragments of megakaryocytes. That is why they are formed elements of blood. Now, one thing I would like to tell here, that just from a few months ago, the question pattern from the examination point of view, the question pattern for at least for the subjects of the, I do not know about the second phase or the third phases, but the question pattern of the first phase of MBBS has been altered, okay, for the first phase of MBBS. And up to the students who have faced the exam up to last year, okay, up to the, for here 2 batches before you, they have faced a pattern of a question which is somewhat modified from for the last 6 months or so, and one batch of MBBS students, that means first phase students, has faced this new pattern. So, there will be one type of question which was earlier also there, just the number of questions and the marks distribution for the questions have been altered, that which will be explanatory type, explain why, okay. So, whenever you read a topic, I would suggest that you prepare the topic for long questions which are of 15 marks, okay, long questions, short notes, explain why, all these types you prepare together. Obviously, otherwise it will get difficult for you. After some days into the course, you just go through the previous year's questions, okay, and try to demarcate this question, this topic has already been discussed, and just try to find answers to those. Otherwise, in 9 to 10 months studying 3 subjects, it will get very difficult if you keep these things for the, that means the last phase means last part of your first phase. So, in the first and there will be 3 internal assessments. In the first and second internal assessment, syllabus will be the in parts. So, whatever is taught from here for the first 3 months or so is the syllabus for the first internal assessment. Then from there on till for the next 3 months is the second internal assessment, but after that you have to face the whole syllabus, that is the preparatory, which is preparatory. The third one is preparatory for the first phase university exam. So, just go through the previous year's questions after some days, after few topics have been discussed. So, you will find explanatory types of questions are there. So, this may be one explanatory type of question, like why the term formed elements is better than the cells, okay, although I have not seen this particular question till now. So, this will be typed, just explain in short. Now, this reasoning, that is explanation type or reasoning type. So, why the blood fragments should be called, blood cells should be better called formed elements. So, this is one example of reasoning. So, you have to just state the reason. So, this reason is actually an one liner. So, these are cell fragments formed for their precursors megakaryocytes, and that is why these are not whole cells. At least one of the components of blood are not, one of the cellular components of blood are not cells as a whole. So, better to use the term formed elements, right. So, and whatever is the description of the structures, we have already done it in the hematology lab. We will be continuing this topic in the hematology lab now, and we will again discuss those and see the cells under the microscope when you are attending the practical sessions. So, coming to the composition of plasma, obviously more than 90% is water, okay, more than 90% in water, and there are so many plasma proteins. Water and proteins compose most of the plasma. What is serum? What is the meaning of the term serum? So, plasma minus, still now we know plasma minus clotting factors. To state very particularly, it should be called as defibrinogenated plasma. So, fibrinogen is one of the proteins in blood, plasma minus fibrinogen, that means defibrinogenated plasma is called serum. So, plasma composition, right, this there will be, from the exam point of view, see from this there will be no long questions, but suppose there is a short note composition of plasma, then you have to write like that. Water, it is mostly water and plasma proteins. There will be some organisms always. So, organisms, bacteria, fungi, microorganisms in there, and there will be the other chemicals, nucleic acids, hormones, okay, all these things will be there, but mostly it is water and plasma proteins. Okay, nutrients, hormones. So obviously blood is carrying the nutrients and oxygen. So blood gases will be dissolved in the plasma. So all this is not very difficult, but mostly it is water. So what is the total? So from here what is pertinent to discuss is total body water and the composition, how much is plasma among that, total extracellular fluid, which will be discussed in general physiology, and very important concept. So just I give you a preliminary idea that total body water, just note it down. Although this is, I repeat, everything is there in the textbooks, but the organization is different in different textbooks. Total body water, it is 60% of the total body mass or total body weight. So this is usually in standard textbooks or in whenever we write, this is written as TBW. So don't just mix it up. TBW is not total body weight, it is total body water. Usually these abbreviations are used. As far as possible, try to avoid using abbreviations, because whenever you use abbreviations, sometimes we mix it up, whether it's TBW is total body weight or total body water, but mostly they are using it as TBW is total body water. Okay. So better for the total body weight, it is better to write TBM, write it as total body mass if you want to write. But whenever you get this term TBW, don't mix it up with total body weight. Don't think it is total body weight. Usually it is about total body water, which is 60% of the total body mass in weight by volume. So in a 70 kg person, it will be 42 liters if it is 60%. If it is a 60 kg, if it will be 36 liters. Okay. So this total body water is divided into extracellular fluid and intracellular fluid. This will be discussed in general physiology. So extracellular fluid, plasma is one of the extracellular fluids. It is not present inside the, what intracellular fluid is present inside the cells of the body, okay, cells. So even the blood cells are the cells of the body, and cells in other tissues. So the fluid inside that is called the intracellular fluid, and the rest is extracellular fluid. So extracellular fluid is plasma and interstitial fluid. Okay. So plasma is only, so if total body water or total body fluids is 42 liters, among that extracellular fluid, which is abbreviated, this is a very well taken abbreviation, this is ECF. Extracellular fluid is only almost, not almost, it is 25, one third, sorry, not 1/4, 1/3 of the this total body fluids is extracellular fluid, 1/3. That is 1/3 of 42 liters. If we take a 70 kg weight, it is 14 liters. Okay. And again 1/4 of this is blood plasma. So extracellular fluid, plasma is one of the types or one of the parts of the extracellular fluid. So except that, the rest of the extracellular fluid is interstitial fluid. So 1/3 of 42 liters is 14 liters, which is the volume of extracellular fluid, and 1/4 of it is blood plasma. Actually it is like a bit less, it is between 1/4 and 1/5. So it's it's like around 3 liters of blood plasma. Okay. So this is a part of the plasma, is a part of the extracellular fluid, and most of it is water, that is over 90%, and among the rest 10%, most of it is plasma proteins. This body fluids composition will be discussed in the classes going on in general physiology. So I just gave an idea that extracellular fluid, what is the part of the plasma in the extracellular fluid. What is the contribution of plasma in the extracellular fluid? That is just 1/4 of the extracellular fluid, which is again 1/3 of the total body water, which is by parts by volume, in weight by volume it is 60% of the total body mass. Better say it, total body mass. In textbooks it will be like written total body weight, but if you use the term TBW, it is for total, it is usually referring to total body water, okay, and it is 60% of total body mass. So this is about composition of plasma. So nucleic acids, you all know, this is DNA and RNA, all these make up a very traces are there in plasma, but mostly it is water and plasma proteins. So functions of blood are very simple, transportation functions, okay, mainly it is the transportation functions, blood gases, nutrients, waste removal. So carrying blood is carried to the kidneys for the excretion function, waste removal, and transport of protective, the protective components, the transport of the hormones from the gland to the areas where they will act, and it is maintaining acid base balance and also temperature regulation. So how does blood regulate temperature? So blood has a high specific heat, okay. So these are physical features of characteristics of fluids and any state of matter. So whatever fluids specific, you will say this better than me, you have recently studied these physical properties in physics, specific heat, high latent heat of evaporation, all these are the high conductivity, all these are the characteristics of blood. Blood is a fluid with a high specific heat, high conductivity, so that the temperature can dissipate. So uniform distribution of temperature from one organ, say one organ to the rest of the body. So organs to the rest of the body, as it is high conductivity. If it has high specific heat, it will require more energy for the temperature to be raised at one particular part. So blood has a role in temperature maintenance of temperature homeostasis. Already you are acquainted with the term homeostasis in general physiology. It is maintaining the internal environment. It is called the internal milieu. It is maintenance of the internal environment within a range where the body functions can be normally carried out. That is homeostasis. So this temperature homeostasis, one of the who contributes to it, blood contributes to it to a considerable extent, because the blood is a fluid with a high specific heat, high latent of evaporation and high conductivity. And all these functions, immune functions we will discuss in immunity, transport functions for nutrients, oxygen, blood gases, not only oxygen, also carbon dioxide, so that the carbon dioxide can be carries from the tissues back to the lungs, so that carbon dioxide can be eliminated from the body through breathing out, by breathing out. So that is transportation of oxygen, nutrient distribution, waste removal, and there are buffering components. One of them are plasma proteins in the blood. Hemoglobin itself is a buffer. So the buffering components which will maintain the acid base balance of the body. So functions of blood are not very, these are simple. So as we told, except plasma, in plasma except water, 90% of plasma in water, but most rest of it is plasma proteins. What are the concentration of plasma proteins? This is the, again see, we just mentioned some parameters will be there which will be there in microliter, some parameters will be there in deciliter. So if we would describe this plasma protein concentration in microliter, it will become a large number. So it is described in deciliter. It is 6 to 8 grams, around 6 to 8 grams total plasma protein, 6 to 8 grams per deciliter of blood. Okay. So all these parameters if we know, so we can convert it into how much plasma proteins in the total body, how will be the total amount of plasma proteins in the body, we can just calculate from this. So in 1 deciliter if it is 6 grams, in 1 liter it will be just 10 times, and if it is 5 to 6 liters of blood, just multiply it by that number, that will be the total plasma proteins. But that is usually not, we do not express it like that, we express it plasma protein standard, the standard practice in expressing it per dl of blood cells in microliter or millimeter cube, blood plasma proteins in dl, hemoglobin in dl, that is deciliter of blood. So 6 to 8 gram per deciliter. These are of various types. Obviously, we are coming to the types, and till now like around 300 have been described. You will, all these you will get in your any one textbook, at least one textbook you have to follow for studying from the examination purpose, and all this information will be there in the textbooks. So they are, what are the conjugated proteins in plasma? Glycoproteins and lipoproteins, which are these conjugated with the lipids, or if it is with carbohydrates, these are called lipoproteins. If it is with lipids, these are called lipoproteins. So plasma proteins are in that way carriers of those carriers. So plasma proteins which carry lipids, they can be called as lipoproteins, and these detailed structures are there, so not detailed, whatever you need will be discussed in biochemistry classes. I think that about these various forms of these nutrients, carbohydrates, proteins, lipids, about metabolism will be discussed in biochemistry. But just about plasma proteins, it is, this is the concentration, this concentration 6 to 8 gram per dl. Remember this is a standard expression, okay, standard expression, and the standard number which you should know. So the plasma proteins in, we all, what you know is connective tissue is derived from embryologically from the mesenchyme. So in the plasma proteins are also derived from, in the embryo, in the embryonic stage it is derived from mesenchyme, and in the adult stage, in the after the life is established and then the individual grows into an adult, most of the plasma proteins are formed in the liver. Some protective plasma proteins are formed in the lymphocytes. They are called, these protective, mainly from the lymphocytes what we get are gamma globulins. So plasma proteins are forms of plasma proteins. We are coming to the forms of plasma proteins. From the lymphocytes in blood we get the gamma globulins, but most of the plasma proteins are formed in the liver, including clotting factors, one of them which is fibrinogen. These are the, this is produced, most plasma proteins are formed in the liver. So liver, in liver diseases plasma protein values will decrease or should decrease. Gamma globulin is synthesized from B lymphocytes. This is one not an exception, but this is one point to note, that these are from lymphocytes and not from the liver. In the embryonic stage, from the mesenchyme, as connective tissue, blood is a type of connective tissue and synthesized by the, it is derived from the mesenchyme. Connective tissue is mesenchymal in origin. We know this. Albumin is synthesized first. We are coming to the forms of the plasma proteins: albumin, globulin. Write down the major forms of plasma proteins: albumin, globulin, and fibrinogen. Maybe along with fibrinogen we can speak of prothrombin. These are the forms of the types of plasma proteins. Which is the major one? That is albumin. So albumin, pre-albumin again is just the precursor of albumin, and albumin, it is albumin which is among the 6 to 8 gram per dl. Almost most of it is made up by, the most abundant is plasma protein is albumin. It is around 3 to 5 or 4 to 5, some textbooks are writing 4 to 5, it is that around 5, almost 5. If it is 8 grams, 7-8 grams per deciliter of blood plasma proteins, almost most of it is albumin. Some part is globulin, fibrinogen and prothrombin are in, by volume it is, or by mass it is much less than albumin and globulin. Okay. Albumin globulin, so ratio like 1.7:1. So in some diseases it is altered. Okay. In some diseases the albumin globulin is altered. In those diseases, whenever those diseases are suspected, other clinical features and all are you are suspecting something on the basis of clinical features and other say laboratory investigations, we go for a albumin-globulin ratio of blood. So, in some diseases it is reversed, globulin may become more than albumin. Okay. So, albumin again percentage of total, most is 60%, almost 60% is albumin. This has become a bit hazy. I will just this right side, albumin is 60%, globulin is like over 30%, and rest is fibrinogen and prothrombin. Some textbooks are writing fibrinogen a bit higher like 7-8%, but this around 5% will do. So, most of it, the takeaway is most of it is albumin, and albumin globulin ratio is reversed. Albumin is almost 2:1, 1.7:1, but it is reversed in some diseases, that is called reversal of albumin globulin ratio, which will be discussed, you will studied in subsequently in your medicine curriculum, that when the albumin globulin ratio is reversed. Okay. So, this is just a recapitulation. Albumins like 5 gram, almost 5 gram per dl, globulins almost 2.5, 2 to 3 gram, and globulins, and again this alpha, beta and gamma, this classification is made on the basis of electrophoresis. So, when electrophoresis, plasma electrophoresis is done, so these are the basically the speed at which this can be separated in electrophoresis. So, they are classifying it as types of globulins as alpha, beta, gamma, and among alpha there are alpha 1 and alpha 2, among beta there are beta 1 and beta 2. Again from this fibrinogen, it is like 300 grams per dl. Fibrinogen is much lesser than albumin and globulin. So, normal serum protein level, as it was in the previous slide, it is like 6 to 8 gram per dl. Just 6 to 8 gram per dl, that will suffice. Okay, that is a standard expression. So, this has, I am sorry, this has become very hazy. There is just variations in plasma proteins. That is, there is a term called hypoproteinemia. Now, whenever you get this term emia, this has become a bit hazy, but do not worry, I will just give you. So, just concentrate on the board, hypo and hyper. Whenever you come across this term EMIA, it is about blood. Okay. So, proteinemia is the concentration of protein in blood. Now, hypo means it is less, that concentration of proteins in blood in fallen, and hyper means it has increased, decreased and increased. Now, there are various physiological and pathological factors for which hypoproteinemia and hyperproteinemia can occur. Like, there are reasons for which in severe burns there may be either of increase in plasma proteins or decrease in plasma proteins. Extensive list you do not require, but just have some preliminary concepts in severe burns. When there is blood loss, so obviously if there is blood loss, a huge volume of blood is lost, and then the liver will take some time at least to, so amount of plasma proteins will decrease. So, that is one cause of huge blood loss, that is called hemorrhage, HEMORRHAGE. So, these are initial days, you will get acquainted with those as your curriculum goes on. In hemorrhage, so plasma proteins will decrease obviously because total considerable amount of blood volume is lost. Again the liver, if the liver is functioning, then it will try to make up for it. So, we have seen in hemorrhage, in burns, in the initial stages there will be loss of proteins, but in the later stages due to different metabolic reactions, there at some point there again may be increase in proteins. Okay. In dehydration, when there is severe water deprivation of the body. So, as there is water deprivation, concentration of various substances in blood rises, and that means what it is occurring, that term will be that can be described as hyperproteinemia, because the concentrations of proteins in that is relative to the amount of water, the amount of substances are increasing. So, in dehydration, hyperproteinemia, one of the causes is dehydration. Now, there are some diseases, the extensive list you not require like this, like it is written hyperproteinemia in tuberculosis, diseases like multiple myeloma. These are some causes of hyperproteinemia which extensive list you do not require, but one or two examples you should know, because from the examination point of view, if you face some short questions or short notes, so that you can write. So, hyperproteinemia, some causes are physiological. In newborns, albumin, still the liver is developing, the hepatocyte cells are developing, it is not as physiologically, it is not same as the adult. So, in newborns, the due to hepatic immaturity, the liver is still developing, hepatocytes are still developing. So, the amount of proteins are somewhat less compared to the what is the adult value. So, excessive loss, severe malnutrition, excessive loss due to burns may lessen the proteins. Again, some diseases in dehydration and some diseases like diabetes insipidus, this multiple myeloma, tuberculosis, proteins disintegrate and increase. Okay. And some reasons are congenital. Congenital means whenever you find the term congenital, what is the meaning of the term congenital? It is by birth, and obviously there will be some or the other reason behind it. Overall, the functions of plasma proteins, if we have to describe separately, that plasma proteins have role in coagulation in blood. As we know, we are describing about fibrinogen, prothrombin. These have role in coagulation in blood. Defense mechanism of body, we usually speak of immunity functions, that is immunoglobulins, which are one type of globulins. These contribute to the defense mechanism of blood. Transport mechanisms, obviously plasma proteins transport so many things. Hormones are binding to them, enzymes are binding to them, and plasma proteins, various plasma proteins carry different substances. So, major role in transport mechanism. Whatever role of maintenance of osmotic pressure, now osmosis, diffusion or osmosis will be discussed in general physiology. Osmotic pressure is the, if you have just a very general idea, diffusion is still now whatever you know, that is substance will move from an area of higher concentration to an area of lower concentration. If it is through a semi-permeable membrane and fluids are present on the two sides of the membrane, if there is a semi-permeable membrane, fluids are present on two sides. If concentration of solutes is more on one side, then the solvent moves from the other side, that means area of lower solute concentration to the area of higher solute concentration. Now, this is called osmosis, but this can be prevented. This movement can be prevented in spite of the concentration difference. This movement can be prevented if certain pressure is applied on that side of the membrane. That is called osmotic pressure. So, maintenance, there is a maintenance of osmotic pressure in blood. A major contributing factor is plasma proteins, because this fluid movement from one side to another needs to be regulated. That means which is the membrane here? Mostly the cell membranes, the capillary walls of the blood. So, these plasma proteins contribute to osmotic pressure, so that in physiologically these movements of fluids across the cell membranes, which is a semi-permeable membrane, is regulated according to the needs of the body. So, role in maintenance of osmotic pressure in blood. Regulation of acid base balance, again plasma proteins, they have biochemically we will come to knowing biochemistry, they have carboxy terminals and amino terminals, and carboxy terminals can buffer H plus, excess H plus in blood, excess hydrogen ions. The amino terminals can buffer bases, so they have role in acid base balance. Okay. So, these are the major functions of the plasma proteins can be described like this. Again, fibrinogen, acute phase protein, you can just ignore the term acute phase for now, just for your knowledge. Acute phase proteins, whenever the body faces inflammation, infections, inflammation, you will study in pathology what is inflammation. Inflammation, infections, these proteins rises, those are called acute phase, when the body is facing some acute crisis. So, fibrinogen, it is one of the coagulation factors as well, it will help in blood coagulation. So, we will study about blood coagulation in a bit detail, the pathways of coagulation. fibrinogen, prothrombin. This is about how the plasma proteins are contributing to coagulation. Again, in the immunity, as we told, immunity, gamma globulins are one fraction of the plasma proteins which are contributing to immunity. There are various plasma proteins, extensive list. You will, as the curriculum goes on, you will get these names one after another. Okay, so many names. You will just come in the text that there are so many plasma proteins. So one of them is transferrin, which will come when on the discussion of hemoglobin. Transferrin transports iron, iron transport in the GI system. Celluloplasmin transports copper. Okay. Transcortin, retinol, lipoproteins obviously binds lipids. Haptoproteins, it is binding free hemoglobin. So if hemoglobin is coming due to some reason, it is outside the RBCs, free hemoglobin is transported by haptoprotein. There is TBG is a very common, which is transporting one of the thyroid hormones, which is thyroxine. So various transport functions of plasma proteins. Osmotic regulation, we told, osmotic pressure, oncotic pressure will be discussed, I think, in the within 1 or 2 classes in general physiology. I just gave a basic idea that for maintaining the regulation of fluid movement across the cell membranes. And plasma proteins have carboxy amphotenic molecules, carboxy terminals and amino terminals, which will help in buffering of the buffering action, which will maintain acid base balance in the body. Okay. Classification of plasma proteins based on their functions, again extensive list, do not go for the extensive list now. So anti-proteases, which are anti-proteases, which are protease are enzymes which are, that means antagonizing these and proteases. So sometimes they are, so that the excess, so that they cannot act in requirement of access. So some of the plasma proteins are anti-proteases. We have already discussed the others, hormones, immune defense involved in inflammatory responses. So those 5 role in coagulation, role in defense mechanisms, role in transport, okay, role in osmotic pressure and role in acid base balance. This is how you describe the functions of the plasma proteins. Again, this is a not very extensive, but at least from one of the standard textbooks in physiology. You do not require to go for the whole list, just these are so many, over 300 have been described till now. Okay, so this is about your basic composition and functions of blood and about the bit of the plasma proteins. We will discuss about the various cells of the blood, hemoglobin, coagulation cascade, blood groups in the coming classes.